I came over to weißensee last summer for the Green Lab project. So my project partner and I were interested in using microorganisms and the potential of pigmented microorganisms to create sustainable dyes for textiles. We hope to find a sustainable solution to the current harmful dyeing and printing techniques and methods used within the textile industry. We teamed up with 2 microbiology students from a lab called TU, and they basically helped us with all the science side of things. They were really amazing. Our aims were to reduce the chemical impact, reduce energy and water use, and to explore cleaner and better technologies. I'll just show you a video so you can kind of get a better idea of what we were doing in the lab, because I can imagine it's quite difficult to imagine because I'd never been in a lab before. There is a huge environmental impact from the textile dyeing industry. More than 8,000 toxic chemicals are used in various processes of the textile manufacture, and the daily water consumption on the in an average size textiles mill is about 1.6 million litres. So we were really hoping to find a way to reduce that. And non-renewable synthetic dyes disrupt photosynthesis, which has an adverse effect on all forms of life: water purification, marine life, soil productivity, and drinking water. Throughout the whole dyeing and finishing process in the textile industry, harmful chemicals are used. 20% of freshwater pollution comes from textile treatments and dyeing. This process is linear. The product can't be returned back to the raw material, or it can't be decomposed without harming the soil due to the chemical processes involved to make the finished product. Natural dyes, so using like plants, fruit and vegetables to make pigments, aren't the solution either. Their colour fades really quickly, their quality is inconsistent, their availability can vary from season to season, and growing them requires vast areas of land. They usually need toxic mordants, which means like to fix the dye to the fabric, they need a chemical. So that's why we wanted to explore microorganisms. There have been projects in the past about pigmented bacteria, but there hasn't really been enough research. So that's why we thought Green Lab was the perfect opportunity to collab with microbiologists. So this is our closed loop material cycle. This is basically how our project panned out. So we start with collecting the microorganisms. Green Lab's brief was to aim to use a local waste stream. So we did focus on soil, yogurt and air to find our microorganisms. They are literally everywhere in the world, and there's millions. Growing them requires an incubator or a fridge, and depending on the organism, we then experimented with designing with the pigments once we'd got them, and using techniques such as shibori, origami and folding techniques, painting and growing them directly onto fabric. Then you have to sterilize it, and then you've got your product. Once you've got your product, this can be upcycled and recycled as many times as possible, because we only use natural monofibers. At the end of the product's life, this is then decompostable, and it closes the loop because it goes back into the soil. The organisms we ended up using were Micro.Colors luteus, which is a bright yellow pigment, and it's found in soil, dust, water, and air. Rototorola rubra is a bright pink yeast, and it's found in soil, water, milk, fruit juice, and air. Lactobacillus casei actually has no pigment until it undergoes a chemical reaction. It then turns blue. It's also found in yogurt and milk. So our first job was to find, extract, and grow these particular microorganisms. Each organism needed different experiments to grow them. We were in the lab a lot and tried many different experiments. They all grow at different rates, at different temperatures, and on different agar, so it was actually quite complicated to keep track of which experiments were happening, all because they were all happening at the same time. This is the process of growing microorganism put very simply. You take a sample from something like soil or yogurt, put it in an agar for it to grow. There'll be many types of microbes which will grow in your Petri dish. You then need to choose a colony to isolate, and then you want to grow it again to make it just be on its own. First, we experimented with collecting soil samples from around Berlin. We brought them to the lab and tried to grow unknown organisms to see what we could get. Many different types of organisms will grow from the sample, and it is quite difficult and takes many different experiments to identify what organism it is. Luckily, somehow we found this one, the yellow one, M. luteus, isolated it and let it grow on its own. So that's what it looked like after we'd isolated it. With the blue one, we collected yogurt samples and did a similar process to the one before. The bacteria has no pigment and therefore look quite white. We spread something called X-Scow and EPTG. I think it's a sugar. Don't ask me the science behind it, but it causes a chemical reaction which produces the blue pigment. But I should make it clear that it is a chemical reaction, but it's not harmful. Like it doesn't produce anything harmful to the environment. With the R. rubra, we really tried to find it. We had so many different soil samples. It was impossible. So the lab provided it, luckily for us. Now we have the pigmented microorganisms. We wanted to know if we could dye textiles with them and if we could design with them. First, we wanted to see if we could get a full coverage of dye and if it would stay on the fabric. We tried mixing the organism in different solutions, water, acetone, and DSMO. Here you can see I'm scraping the organism out of and putting it into one of the solutions. They're then left to shake for a couple of days, and then we added the fabric into that, but it was unsuccessful. As you can see, it just didn't dye at all. It was too diluted. Next, we wanted to see if we could extract a pigment in both a liquid and a powder form. It did work, however, it was in such small amounts, the colour isn't strong enough to even try on fabric. So then we realised that we would need to use like the whole plate of the organism without diluting it if we wanted a strong colour. We tried different methods to see if we could design with the microorganism. And yeah, here's what we found. So this one, we pipetted the organism straight onto the fabric to then let it grow on the plate to see if it would come up with like some sort of pattern, if it would be like full coverage. We had no idea at this point. We tried like different types of yarn, scrunching it, tying fabric, folding, clamping. And this is what they look like in the Petri dishes. So that was the yellow microbe, the pink one, and the blue one. As you can see, like it kind of looked a bit tie-dye once it came out, once it was sterilized. Next, we tried massaging the organism straight onto the fabric surface. This was basically just to see if it would, like, once sterilized, stay on the fabric, if it would dye it. It did work well. We did get full coverage. And it was quite bright as well, so we were really happy to find that out. That's what it looked like after sterilization, so it was safe to use then. The color did dampen a bit, but yeah, we were just happy we'd finally found something because this was a few months into the project. We then tried a few like origami and shibori techniques, because obviously we didn't have that much organism to use onto fabric samples. We wanted to get bigger samples with some sort of design methods. But yeah, that's why we tried these kind of techniques, which are stamping, rubbing, wrapping. So yeah, we basically found after all our research that 1 litre of water can dye 4.6 meters squared of fabric, and no chemicals, mordants or textile treatments are used within the dyeing process. If the process was scaled up to an industrial context, there is a possibility of extracting a large range of colours from locally sourced microorganisms, minimising water use and chemicals in the textile dyeing industry. Here are some of our final samples. We've also got an archive of all the samples and what like scanned in if you wanted to have a look.